All right. So one reason, well, there are multiple reasons why marine mammals might be of particular interest to study when it comes to looking at plastic pollution or ecotoxicological effects in general. And that's because a lot of marine mammals act as sentinels of marine ecosystems. A lot of them are often keystone species as well. Um, Most of them are higher trophic level, or they're feeding on large volumes of um, like a large biomass of food. If you think of whales filter feeding, they're eating a lot of krill, they're encountering large volumes of water, potentially being impacted by higher concentrations of perhaps plastic particles, for example, or other organisms higher up the food chain. Like today, I'm going to be talking about elephant seals might be vulnerable to things like persistent organic pollutants or plastic additives as a result of their diets. So another reason why they're also interesting to study is because marine mammals are vulnerable to other anthropogenic effects as well. Sorry about the sirens. Um, and so they give us a really good way to see how ecosystems are doing that we might not have as easy access to just because of where they are and what they might eat. So in my research, I specifically focused on the northern elephant seal as a model system. The reason for this is because they're pelagic and deep divers. So northern elephant seals forage off the coast of California, kind of up near near Alaska and further offshore as well. They're known to forage on things like myctophid fish, like lanternfish and squid that have been found to contain plastic particles and additives. The other thing about northern elephant seals is that they spend 90% of their lives out at sea, and they come back to land twice a year to breed and to molt. So unlike some of those other pelagic marine mammals like whales and such, northern elephant seals are easy for us to sample. They also have large fat stores, which make them a primary candidate for looking at things like plastic additives or persistent organic pollutants, perhaps in future research. Here's a map of female northern elephant seal foraging, as well as a map of microplastic concentrations at depth. So I mentioned elephant seals feed in pretty deep areas. Females dive particularly like 300 to 800 meters. You can see that on the right. And the graph on the left is from a paper showing microplastic concentrations at depth. And this is in the Monterey Bay National Marine Sanctuary. So a little bit closer to shore, not necessarily exactly where elephant seals are foraging, but as I'm sure a lot of you are familiar, plastic concentrations at depth are a little harder to come by, but it's interesting to see that perhaps elephant seals are being exposed to higher concentrations of plastics than we can deduct from surface waters, for example. So they might give us an idea of these plastic concentrations at depth, their composition in ecosystems that we're not as familiar with. So my research questions for my projects were, one, are northern elephant seals consuming microplastics, considering the depth at which they forage and the things that they forage on? It seems like probably they might consume plastics. However, northern elephant seals are really well studied, but no one's looked at plastic pollution in them until this. Next, if elephant seals are indeed being exposed to plastic particles, how might those impact a northern elephant seal? So if you imagine an organism consuming a piece of plastic, those small particles likely come into contact with other cells, either along the digestive tract or elsewhere within an organism. So we were curious to see how those plastic particles might impact northern elephant seal cells in particular. And then lastly, we're interested in how plastic additives might impact northern elephant seal tissue physiology. As I mentioned, plastic additives, in this talk, I'll be talking about BPA and BPS, are lipophilic, and northern elephant seals have large fat stores. BPA and BPS are also known as obesogens, and northern elephant seals are naturally obese. So it would be interesting to see how these additives might impact their like blubber tissue or adipose tissue physiology. So the first thing I wanted to figure out was, okay, are northern elephant seals exposed to microplastics at all? And the way we decided to go about that is to quantify microplastics from northern elephant seal scat samples. To do this, we collected scat off of the beach from Point Reyes National Seashore and Año Nuevo State Park, just off the coast of California. We collected 8 from Año Nuevo and 3 from Point Reyes. At each sampling location, I also took samplings just to ensure that any potential particles that I found in my samples were a result of something that came out of a seal rather than was just environmentally around, like on the beach. And these were collected during their spring and summer haul outs. So what I did was, once I got my scat samples back to the lab, I weighed out subsamples so that I can later find concentrations of plastics and to control for the different sizes of scat or potentially size of animal that these scat may have come from. But then I performed a density separation where I mixed my subsample in a hypersaline solution, allowed that to settle overnight. Then the top layer of that, after that density separation, is hopefully contains just synthetic material, but I use a serological pipette to pipette off that top layer, move it to a new beaker, and start a chemical digestion just to digest away any excess organic material. Oops. Okay. Then I used vacuum filtration system to end up with filters that look like this. And with those 11 samples along with blinks. Sorry, I got a question in the chat. I don't know how distinguishable the scat are between the different pinnipeds, but in the areas we were sampling, they were just elephant seal rookeries, so there wasn't anybody else around. Okay. Yep. Oh, I ended up with filters that look like this. So I ended up with 100 filters with those 11 samples, including my blanks. I took 3 photos randomly of each filter to account for 33% of the filters themselves. And my photos ended up looking something like this. While I was doing all the laboratory steps, I also used filtered water to do laboratory blanks to account for any laboratory contamination. And then on the bottom, you can see an example of a scat filter from Año Nuevo. So my question was, are northern elephant seals consuming plastics? To figure that out, I multiplied my filter counts by 3 as I took pictures of 33% of my filter, divided by the grams of the subsample. And then because my data were non-normally distributed, I used a Wilcoxon ring sum test, which is just basically a non-parametric t-test to compare the plastic concentrations in my scat samples compared to the combination of my lab blanks and samplings. And based on our data and that low p-value, we would say, yes, it does seem like northern elephant seals are consuming plastics. Then we had this bonus question. If I mentioned earlier, I sampled northern elephant seal scat from 2 different locations. So we were curious if microplastics concentrations may have varied by location. To do this, I corrected the scat concentrations by both lab link average concentrations and SAM-blank average concentrations from each particular sampling site. So I had Año Nuevo lab corrected concentrations, and I subtracted Año Nuevo SAN concentrations to end up with this overall corrected concentration that I could compare between the 2 sampling sites. I used the same non-parametric t-test as my data were still not normally distributed. And I ended up with concentrations oops. Oh, I couldn't see it. I ended up with these different concentrations between Año Nuevo State Park and Point Reyes, where it looks like there are higher concentrations of microplastics in my samples from Año Nuevo compared to Point Reyes. So just to visualize that a little bit, a reminder here on the x-axis, I have concentration or particles per gram. So the further to the right a particular curve is, the higher the general concentrations, and the more to the left, the lower the concentrations. So here, you can kind of see on the right, there's more orange than purple. So that's just further showing that Año Nuevo samples tended to have higher concentrations of microplastics. So we were wondering why that might be. It seems like elephant seals from Point Reyes and Año Nuevo contain different concentrations of microplastics. So we first sought to see like maybe concentrations in surface waters near both of those locations might be different. So we searched the literature for that. And it seemed like concentrations in Santa Cruz had higher concentrations of microplastics compared to further up north near Point Reyes. So maybe that could be a reason that Año Nuevo seals might be exposed to more microplastics than Point Reyes seals. But we were, to further control for potentially these two different rookeries, we did also compare sand blanks from both of these locations as well. And we did not find a statistically significant difference between microplastic concentrations in the general land environment that these seals were coming from. Another reason, and this is kind of what we're more thinking of what might have driven this difference, is that we sampled our scout at two different times of the year. We sampled from Año Nuevo during April, in which there were adult females and juveniles on the beach, and we sampled from Point Reyes in August when there were only adult males on the beach. So perhaps there was a difference by sex. And the reason this might be is because males and females forage in slightly different locations. So here you can see the males forage more along the coast near the Aleutian Islands, and the females in red forage more towards the North Pacific. And since we think likely microplastics exposures are likely coming from their diet, it would make more sense to us considering these elephant seals basically stop foraging somewhere out at sea before they return back to their rookery. We think perhaps these differences in foraging locations might be driving these differences in concentrations, but we can't know for sure. So here's a couple other maps. So we have the same one on the right with male and female foraging. And on the left, you can see a graph of surface water microplastic concentrations, where the warmer tones have higher concentrations of microplastics. Just as a reminder, these seals feed at depth, so we're not really sure necessarily how well these concentrations correlate back and forth. But if we're just looking at surface waters and that's any indication for what's happening at depth, it seems like perhaps female elephant seals might be more susceptible to microplastic exposures based on previous microplastic presence studies in the same general areas that elephant seals forage in. So so all in all, the concentrations of microplastics that we found in these seals are actually likely the minimum concentrations that these seals are exposed to, since I mentioned they probably finish foraging offshore before they make their way back to their rookeries twice a year. And we're unsure of the gut transit time for these seals. So likely an elephant seal may have already defecated plastics that it may have consumed further out at sea, and the samples that we're collecting may have just been any residual plastic that had been left in the guts of, or the digestive tract of these elephant seals. Additionally, previous studies have shown that really small pieces of plastics, like nanoplastics, can translocate. So perhaps there might still be more plastics within the organism that we can't really test for from live organisms. Okay, so there were a few limitations to this part of the study. The first was, you'll notice there's a relatively low sample size. It's kind of the nature of sampling from marine mammals as one drawback. It's pretty cool that it looks like there might be a difference between location, but since we sampled one sex from one location and one sex from another location, it's pretty hard to disentangle those confounding variables. So it would be interesting to study both sexes from one particular location in the future. Next, it seems like since these elephant seals seem to contain microplastics and we think they may be coming from what they're eating, it would be interesting to see what the microplastic concentrations at depth are in these ecosystems, as well as presence of microplastics in the particular fish from the ecosystems that these elephant seals forage in. Similarly, we have a lot of tracking data from these elephant seals, so it also might be interesting to model perhaps what risks elephant seals might have to be exposed to microplastics as well. And lastly, with these filters, I have not done this yet, but we're planning on running FTIR spectroscopy just to better link to confirm that what I counted are indeed plastics and to figure out maybe what types they are. It might give us an idea of the composition of plastics at these depths where they might be foraging. Also just in general to help us better link what we found to an original plastic source. Okay, so to conclude this part, it seems like elephant seals are indeed consuming microplastics. In my scat samples across potentially 11 seals, but we'll just say they were 11 scat, 100% of them contained plastics. And second, their exposure may vary due to sex, but more research is necessary to figure this out. And then the next part is, okay, if these seals are exposed to plastics, how might those plastics particles be impacting them? So the way we sought to look at this is using a cell culture model. Cell culture is pretty commonly used in looking at ecotoxicological studies, as it allows us to test different sizes, different concentrations, basically just different treatments and how they might affect a particular cell, especially when it's not feasible or ethical to sample, to use live animals to test the effects of something. This is often the case in mammals, specifically marine mammals, because we didn't just go out to the beach and feed seals a bunch of plastics. Also, previous studies have shown smaller particles at higher concentrations tend to have higher toxicity. So we were curious to see if we would see a similar trend in our seal cells. Okay. So the way we went about this was we isolated skin-derived cells called fibroblasts. The reason we chose them is because they're robust and relatively easy to isolate considering they're primary cells. So there aren't any cell lines that I know of for marine mammals. And this is a baseline study in vitro. So we just wanted to see how will plastics affect elephant seal cells in general. So the first thing we did was we collected biopsies from these seals on the beach. We took this top skin portion and grew them in culture. And we ended up with cells that look something like this. So these are fibroblasts. Then I exposed half of them to a particular treatment. In this case, we used polystyrene of 0.05 microns at a 1 to 10 dilution of what the original bottle came with. So for this, we're using virgin microspheres and exposed cells for 24 hours. These cells were grown on slides and stained, so we can see maybe how morphologically the cells, like maybe the cells look different as a result of being exposed to nanoplastics. And what we found was, so these are two photos of my microscopic images, my stained fluorescence. The red lines are actin filaments, which basically lie underneath the cell membrane and give us an overall picture of what an individual cell might look like. And the blue dots are genetic material or nuclei. These pictures look quite different from each other, but what we're focused on is individual cell morphology. So if you look at the way the red lines are spread out between the left picture and the right picture, you can see that those don't overall look very different. Fibroblasts, when they die, tend to ball up. So if we saw a difference, we might expect some of the lines on the right side to be balled up in some way or tangled. That would look different from the nice spread out versions that we see here. So it doesn't look like we see a different in morphology. And we did this across a couple slides as well. These are just a couple of representative photos. We were then interested to see if, okay, maybe the cells look okay, but maybe their viability might be different. So to do this, we set up these MTT assays. So on the right is just a figure of what a particular plate might have looked like when I ran these assays. I ran my control groups, the negative control with no plastic exposure and no hydrogen peroxide in 6 replicate wells. And then my positive controls and treatment wells were run in triplicate. I used positive controls of 5 millimolar and 10 millimolar hydrogen peroxide, as we knew that hydrogen peroxide should decrease cell viability. So if I ran this assay and the cells in those wells did not look great, we would probably go ahead and say that the assay is working. But then in my treatment wells, I used 2 different concentrations at 2 different sizes to see how they might be affected. So the way this assay works is I first, after that 24-hour exposures to treatments, I took off all of the plastics, all of the hydrogen peroxide, and then I added this MTT reagent, which through mitochondrial reductase, if cells are happy proliferating or metabolizing well, they'll take up more of that reagent and then form these formazan crystals. So that's what these purple lines are. And this assay is a colorimetric assay. So then I was able to dissolve away those crystals and ended up with basically these different colored wells that I can run on a plate reader. So we'll see the higher the absorbance, the higher viability. So in a couple of my experimental runs, it did look like cell viability was impacted as a result of being exposed to these nanoplastic particles at the different concentrations. These are just 2 examples of that. What's really nice to see is that our MTT assay seems to be working well. You can see, like in my control groups, going down the different concentrations of hydrogen peroxide, we see an increased or we see a consistent decrease in cell viability, indicating that our assay is working with here relatively low variability. It also seems like overall, in general, our error bars are all lower than that control group in our compared to our treatment groups, compared to our control group. Sorry, the treatment groups look like they're not doing as well compared to our controls. However, after doing this experiment several times, I ended up running this assay about a dozen times, which took several days. I actually, this was months of coming in every day to check on cells and run these assays and scheduling it out, testing different concentrations and such, and different sizes, as you can see on the x-axis here. I ended up not really seeing a consistent trend. So it was really cool in a couple of our experiments where it looked like maybe there's something going on. But after running these assays a few more times, it seemed like there was a lot of inter and intra assay variability. So this graph represents 6 replicate experiments. So as I mentioned, I ran this assay several times. This is just representative of times of using particular treatments. So as I mentioned, I tried a lot of different sizes at a lot of different concentrations, but the ones that were consistent and the ones that we tended to have stuck with based on how much we had left and just how the earlier experiments were run were the 0.05 micrometer and 0.5 micrometer polystyrene beads at the 1 to 10 and 1 to 100 dilution. You can see there are pretty high error bars here, but we did still see that decrease in viability as a result of the hydrogen peroxide treatment. I also wanted to point out that we also ran a well with no cells in it at all that I exposed to plastics just to make sure that maybe if I didn't rinse away enough plastics or whatnot, that wouldn't cause any background absorbance readings. So we felt good about how low that was as well. So we're not really sure what's happening with the high variability here. Growing cells in a 96 well plate, you know, I counted them out before I plated them, but I can't guarantee the number of cells per well. And so that could explain some of this variability. I also wanted to point out, since I'm giving this talk to quite a few people from OSU, so I wanted to shout out the fact that we did try to do this with weathered PLA particles as well. And based on our results from running this assay, you can see the control PLA. I just arbitrarily assigned this to 0.7 on the right. All of these bars here, the control PLA and weathered PLA particles at the different concentrations didn't seem to have an effect on viability, at least in this run. And you could also see that these error bars and general overall mean absorbance was kind of similar to control, where it looks like the cells didn't seem to be affected as a result of being exposed to these particles. So there were also limitations to this as well. This is a common theme with, again, marine mammal work, is a low sample size. For this particular experiment, to maximize the yield of cells that we got, we pooled biopsies from different seals together. And so what ended up happening was we didn't actually end up with true biological replicates. So we were limited in that we couldn't perform statistics on these analyses, but just based on the high variability, we can't really draw any strong conclusions as to what might be happening here. Next, I mentioned that we use some weathered plastics. In this case, we use PLA. I think it would be interesting to run this experiment more times with potentially the same plastics or a different weathered plastic to see how those might affect these cells. As northern elephant seals or just animals in general are likely not necessarily being mostly exposed to virgin, perfectly spherical plastics, but I still think it's interesting to see, considering we wanted to kind of disentangle the potential different additive effects by just looking at specifically the plastic particles themselves. And lastly, perhaps cell type may have been something to do with this. Fibroblasts aren't necessarily being exposed to plastics in real life. We just use them because they were easy to access and relatively easy to grow. But in the future, it might be interesting to look at peripheral blood mononuclear cells, as in the past, those have exposures to plastics have shown effects in oxidative stress, which is also an assay that we try to run as well. But the kit that we bought just didn't agree with our cell type. So maybe someone in the future could work on that. So it seems like in morphology, it doesn't look like the cells are being affected. We think that maybe growing cells at the bottom of a well with plastics in the well might mean that the cells and plastics aren't necessarily interacting with one another. So in the future, it would be interesting to confirm this interaction via microscopy, perhaps buying fluorescent particles instead. That could give us a better idea of how much the cells and plastics are actually interacting. If they're truly not, it potentially we could grow cells on slides, float them to the top of a well, and flip them over to just maybe give more of a chance for plastics and cells to interact. But those are just a couple of ideas there. We didn't see consistent effects on viability, but I like to think that maybe the cells are like surviving but not thriving. So there could be sublethal effects that we're not necessarily measuring by just looking at viability. Okay. And then, sorry, the last part of my talk is looking at the effects of plastic additives in northern elephant seal blubber. So those obesogenic additives, BPA and BPS. So most BPA comes from land-based sources like factories, and they're commonly used additives in things like receipts, water bottles, canned food liners. They're used when making polycarbonate, and they also tend to, BPA and BPS, when they adsorb, they have high affinity to polyamide and polyurethane. The reason why I point this out is just because previous studies have found polycarbonate and polyamide are some of the highest concentrations, the highest proportion of microplastics at depth where these elephant seals might be foraging. But in waters offshore where the elephant seals are, it seems like there are relatively low concentrations of plastics. But seals have been found to contain these additives as a result of their diet. Okay. The way these additives potentially work is by acting as endocrine disruptors by mimicking estrogen. They can bind to estrogen receptors and thyroid receptors, which are nuclear receptors that might alter gene expression, leading to downstream metabolic effects like obesity and insulin resistance. So to test what might have happened as a result to BPA and BPS exposure, we focused on this adipose tissue. This was done by some of our collaborators from Belgium. They came here and did this field work, and they exposed these precision cut pieces to different treatments. So epinephrine is a lipolytic hormone, meaning it breaks down fat, and BPA and BPS are thought to be obesogenic. So we were interested to see how those two interacted with one another as well as on their own. To do this, I did RNA extractions on these different exposed tissues, did sent our samples for RNA sequencing, assessed our quality, found out that 85.3% of the genes that we expect to see in a typical mammalian transcriptome we found in ours. So we felt good about that, considering we have a northern elephant seal, which is not exactly a model organism. And then we just clustered, allowed our samples to cluster based on global gene expression. And we found out that it seems like these clustered based on individual seals. So we had to do some statistics and blocking to make sure we were comparing the effect of the treatments rather than the effect of individual seals. Then I annotated our differentially expressed genes. It seems like the contaminants themselves didn't greatly alter gene expression. We had low numbers of differentially expressed genes. This matches with our PCA that I saw earlier, where all of these genes are kind of close together, meaning they're not very different from one another. In the combined treatments, so for comparing the lipolytic hormone with contaminant versus just lipolytic hormone, there's only one differentially expressed gene, suggesting that a lot of these changes are being in these treatments compared to control are being driven by epinephrine rather than the contaminants themselves. However, these numbers on the far right are uniquely differentially expressed genes. Though we saw those low numbers of differentially expressed genes, it is interesting that we found that those genes that we found were all involved with lipid homeostasis in some way. The reason why this is important is because elephant seals have a feeding fasting lifestyle that heavily relies on lipid homeostasis. Basically, while elephant seals are out at sea, they're feeding and building fat stores, and they need insulin for fat deposition. So they need to be able to create these large fat stores. But while they're on land, they need to be able to break down those fat stores in order to do those important activities for fitness, including breeding, molting, nursing, and things like that. So if these genes are being differentially expressed and impacting these elephant seals' ability to do those things, then that could potentially not be great for elephant seals in general. So just to conclude, it seems like we see microplastics in northern elephant seals. The cells don't look like they're impacted, but further research is necessary. And it seems like BPA and BPS, though they don't greatly alter expression, do alter expression in genes associated with lipid metabolism, which could impact their overall fitness. So overall, marine mammals are a really great system to look at these things in. I don't have to tell all of you the importance of studying plastic pollution. And yeah, I just wanted to thank my lab. I want to thank the SEALs, all of you for listening. Sorry, that was longer than I thought it was going to be. And yeah, thanks for listening. I'm looking forward to hearing from you all.
Thank you so much, Lauren, for sharing your work. Really an impressive amount of work and lots of different tools that you learned and applied. So congratulations on completing your master's. And yeah, I guess let's open up the floor to questions. I'm sure I see the chat has been active during your presentation. Lots of thinking about maybe why you were seeing more plastics in some parts in the one population versus the other. Does anyone have any questions for Lauren? Paul, go ahead.
Hi, Lauren. First of all, great project, and thanks for your presentation. It's really fascinating. These animals are really fascinating. I'm really fascinated by the poo aspect of this. So I can imagine collecting poo was very difficult, but has anybody collected their prey items and examined the prey items for plastics, and then just perhaps you could scale that up to a more accurate more accurate estimate of sort of their exposure to plastics and consumption of plastics.
Yeah, I think the previous studies that I was mentioning about plastic pollution in those myctophid fish are mostly in the Atlantic rather than in the Pacific, where these elephant seals are feeding. So to my knowledge, I don't think that we know exactly in the Pacific what plastics they might be eating. Also, we don't have as clear foraging data about the adult males. So we think that they feed on slightly different things, but we're not totally sure there either. So it would be hard to know exactly what to sample for them.
Yeah. Okay.
Yeah.
If you're interested in collecting samples from fur seals, I have a colleague who does research in Alaska on fur seals. So if you want to continue,
yeah,
put you in touch. But great job. Thanks.
Thank you.
All seals all the time. Leah, go ahead.
Yeah, I just wanted to say great talk, Lauren. I was curious when you were doing your primary cell culture, if you noticed, one, I totally sympathize because I've done primary cell culture before and it can be a beast troubleshooting it. I was curious if the particles themselves, if you were able to like see them interacting with the cells or at least like up against them, because I know like buoyancy can be an issue even like in the individual wells. So I was just curious if you noticed anything anecdotally.
Yeah, I couldn't see. I guess when I was looking at them, I couldn't tell if they were interacting at all. Just like the depth perception, I guess. So that's why I think fluorescence would have been interesting to look at there. There was something else about that. Oh yeah, in terms of them interacting, it would have also been interesting, I think. I mentioned the like flipping the slide over thing, maybe also just using potentially just more dense plastics that are like not as buoyant in media. But yeah, I didn't see or notice anything. No, I couldn't tell.
Yeah. The other thing I was going to mention, because this is more of a comment and it might be like helpful just moving forward if you ever, if you haven't been burned too badly by primary cell culture. I noticed when I was working with the cells a lot, my cells, nothing in seals, this was in fish, did not adhere. They didn't actually stick to the bottom of the wells. And so whenever I would do any kind of like washing step, I would lose a ton of cells. And that created a lot of variation in my wells. So that might be if you have like a lot of washing steps and and you're not sure if your cells are like really sticking to the bottom, that might be the big source of variation. Because I noticed even in your controls, there was like a really big error bar. So I don't know.
Yeah, I tried looking at them between wash steps too, but like I didn't count them at that point.
That's interesting that you mentioned that because we're trying to get some Manidia cell lines going here, and we're having the same challenge with floating cells. Maybe that's just a thing fish cells do.
Yeah, I was doing kidney cells, and it was supposed to be like predominantly macrophages, which, if you have a cell line that's macrophageous, they should stick really nicely to the bottom. Mine never did that. I'm sure that you could play around with every environmental factor to get them to do that. But I basically figured out that I had to, rather than like vigorously washing them like you would do in a cell line, I would remove most of the volume very, very gently and then very gently add, because they would settle to the bottom, but yeah, they would never stick. But I did, if it's helpful, I did write a paper on all of this, and it's a lot of tips and tricks because it's in Methods X and it's open access. So I'll drop it in the chat. My hope was, I was like, no one will ever read this paper, but if it has a nugget of knowledge that helps someone get their project off the ground, that would be the dream.
We will read it and cite it. That's all. Thank you. Sarah, it looked like you had your hand up a minute ago.
Oh yeah, I was just gonna ask how she dealt with the floating cells because I'm working on the project and the main problem is the floating non-adherent cells. So that's good to know.
It's good. It's good to know that it's not just us. So, cool. Lauren, about the cell line work, do you think in the future, I'm not sure if there's plan to continue, there probably is plan to continue work on elephant seals down at U of P. Do you think other cell line types might be pursued, or would there be any potential opportunity to try to immortalize one of the cell lines? So it's something you could sort of keep working on and have it be more consistent. And I know those are all complicated things to do, but
Yeah, I think they were, well, I don't know if people or this lab is going to continue down the line of plastics research. I know one of our collaborators, in terms of the additives, that might be something that continues to happen, but plastic contaminants in cells themselves, I don't know. I think we talked about maybe doing using blood, for example, but it just seemed not necessarily like the easiest for something that's baseline. So I don't know if they're going to do something like that in the future. I think it would be really interesting. But our lab has tried to like immortalize seal cells before, like from fibroblasts specifically. And I think they got to passages like 15 or something, but couldn't quite keep them. And they also didn't look super great. So I don't know if there are any plans to try in a different cell type at all.
cool. Yeah, and I realize getting access to those tissues is difficult when you're talking about an elephant seal too. I was wondering, does the lab collaborate at all with the Marine Mammal Center in Marin County?
Yeah, we currently don't, but my lab mate and I and our PI have all volunteered there in the past. There was some talk about potentially doing some things with with like delayed implantation, for example. So they wanted to use like captive animals to do that, or deceased animals for preliminary data. But there was some political stuff, as the marine mammal world can be. So I don't know if there are plans for doing stuff in the future, but no, we don't currently collaborate with them.
Just it came to mind. I used to volunteer there briefly before grad school when I lived in that area. Um,
Yeah.
I was on the Friday night crew.
On
But
Thursday day.
does anyone else have any questions for
Yeah,
Lauren? We have a few minutes left. Go ahead,
I have
Ariadne.
a question. Okay, so that was a great talk. Thank you so much, Lauren. I'm really looking forward to seeing the recording actually, so I can, because I feel like sometimes I'm a little slow. My question is, did you, when you were looking for plastic in the scat, did you happen to find other, what you might think of as man-made materials as well, or even did you find food? I'm just curious to see how that part of the experiment went.
Yeah, I think I can't tell for sure like what I necessarily found. So I think the next like FTR steps are going to be really important. But I did denote but not do any stats on or count or add together fibers, and it seemed like there were quite a few. But in terms of like other things, I don't think I saw any food or anything. I surprisingly didn't come across any worms. I don't know if they just like got digested away. Um, but yeah, so I'm not totally sure. It just seems like potential particles, and the fibers were pretty clear. Mhm. Thanks.
Kim, go ahead.
Hi, thank you so much for that talk. That was really cool. I'm really excited to hear another project working on microplastics in marine mammal feces. So a question for you, a couple of questions. One, I think you said you ended up with about 100 filters from 11 fecal samples. Is that because the filters were clogging pretty quickly? And did you do the hydrogen peroxide oxidation as well as the density separation?
I did a potassium hydroxide
Okay.
digestion. And then I think it took there's a couple reasons the 100 filters happened. I had to test these methods quite a few times. So for a couple of subsamples, I just use them to figure out the methods to do this exactly. Since elephant seals, we don't really know about plastics much until this. And then the rest of the filters I used for like lab blanks and sand blanks. It also still seems like a lot of filters, but it was 11 scat samples, but several subsamples. So that also accounted for more, and some samples more than others clogged a bit.
Okay. And so when you said subsamples, did you do any biological replicates? And were you able to compare um, the number of microplastics isolated from replicates subsamples of the same fecal sample?
I did not compare between the subsamples within one particular scat. No, I didn't. But I guess I could. I have all the data.
I'd be really curious to see. So we've isolated microplastic particles from killer whale feces, and we just see a large amount of variation, both in terms of how they're distributed throughout the scat. So there's variation even at the level of the individual subsample, which makes it a little bit challenging to identify significant patterns. But it is interesting, and I'm sure that the consistency and the globs of lipid and things like that are probably playing a role there.
Yeah, no, that's a really good point. Yeah, thank you. I'll definitely look into that since I can.
This is really cool. Thanks.
No, so are killer whales.
And then Jennifer, who is in our lab group, she's a master's student. She's not here today, but she's looking at microplastics in otter scat. So she has samples from Alaska and samples from the Seattle Aquarium, and I think she went through just as many filters per sample as you did. Just, I mean, a lot of it was like shells and rocks, and just it takes a while to troubleshoot too. So yeah, similar experience here. But yeah, any other questions from the group? We probably have time for one or two more. No. I just kind of had a question about sort of basic elephant seal, foraging, and what, and I may have missed it, but why is it that the females swim so much further offshore, and what is different about their diet in comparison to the males that they're swimming that what looks like to be a much further distance potentially to obtain food?
Yeah, I don't know. I don't know what's driving that. I don't know if somebody does, but I think it's thought that females feed mostly on more on myctophid fish than males that might feed more on squid. But
Mhm.
other than that, I'm not really sure. I don't know if maybe like males just because they're so much bigger require like a bunch more and maybe there's more availability in that, but I don't know why that would cause females to not to go farther.
Right.
Yeah, so I'm not sure.
The males aren't too friendly. Maybe they're trying to get away from the males for a little while, but no,
Maybe.
for now. Great. Well, thank you so much for your presentation today. Really interesting. And again, like I was saying, really impressive collection of different approaches too. And it seems like there's a lot of potential to build on this model now that you've created these different tools. So if no one else has any questions, can finish up for today, and we'll be meeting again, slightly different group, but some of you are in the same group next week to talk about the tire particle synthesis paper next Wednesday, and then we'll continue the monthly effects group journal club meetings on the third Wednesday of every month, at least for the rest of the academic year here. So if anyone has something they'd like to present in April, or if you have a colleague who wants to present to the group, just shoot me an email and let me know. But yeah, give Lauren another round of applause for a great presentation. And we'll see everyone next month, or potentially next week.
Suzanne?
Yeah.
Suzanne, can I? Yeah, hi. I just want to put in a plug for next week's meeting. I'm really excited about the response that I've gotten from folks about attending that meeting and getting involved in it. And I just want you all to know I've been working feverishly on a draft of the paper, and I've made progress. And I've made progress because I've borrowed very liberally from Kelly Moran's paper and from another EPA document, unpublished document. So I'm excited about next week, and I'm really looking forward to meeting with you all.
Great. Awesome. Sounds good. We'll see you next Wednesday. Thanks, everyone.